Welcome everyone to this My Macular and Me webinar. It's lovely to, well, not to be able to see you all, but to know that you're all here again. This is a really interesting topic. Could plants be used to treat macular disease, which I think is such an interesting idea. And we're delighted to welcome two really special guests from the University of Surrey, Professor Dulcie Mulholland and Elisha Griffin, to talk about an exciting area of research that explores the potential of plant compounds in treating macular disease. Before her recent retirement, Dulcie was Professor of Organic Chemistry at the University of Surrey and has had a long-standing interest in natural products and their medicinal potential. She'll be joining us for the Q&A at the end, but the main talk this evening will be led by Elisha Griffin, who is a Macular-funded PhD student who has been working closely with Professor Mulholland throughout her studies. Elisha grew up on the South Coast and graduated from the University of Surrey with a master's degree in chemistry in 2021. And it was during her final undergraduate project that she became fascinated by homoisoflavonoids, which are naturally occurring compounds found in certain plants that may have potential as new medicines. She's since spent the past few years researching how these compounds might help prevent abnormal blood vessel growth in the macula, and she's now in the final stages of writing up her PhD thesis. So we're really thrilled to have her here tonight to share more about her fascinating work. So I'll be handing over to Elisha in a moment, who'll talk for a bit about her research, and then we'll have a Q&A session. So remember, please do post any questions that you have in the chat box, and we'll do our best to answer them after the talk. So I think I've covered what I need to in the introduction. So now I'll hand over to Elisha. Fantastic. Over to you.
Thank you, Ed. I'll just share my slides. Is everyone able to see that?
Yes, that's perfectly visible.
Fantastic. Thank you. So, hello everyone, and obviously thank you, Ed, for the introduction. And thank you all for joining the webinar today to listen to my talk. So today I will speak a little bit about my PhD research project that has been generously funded by the Macular Society, which simply has explored whether compounds from plants and compounds inspired by plants could lead us to the next treatment for macular degeneration. First, I just wanted to introduce myself just a little bit more. I know Ed already said I attended the University of Surrey and graduated in 2021. I've always had a passion for medicinal chemistry. If you don't know what that is, it basically involves the chemistry of designing, testing, and taking new drugs to market. As Ed said, Professor Mulholland was my supervisor during my final year undergraduate project, where I was introduced to the potential of plant-based compounds to treat macular disease. After this, she told me about the grant from the Macular Society to continue her group's research and urged me to apply. So now, 4 years later, the project has come to an end. And during this time, we've presented this research all around Europe, and I was lucky enough to be able to go on international research placements. We're currently in the end stages of the project, and we'll be publishing to scientific journals in the next few months. So that's how I arrived here today, but what about the research? So I'm sure a lot of you here today are very well clued up on macular degeneration and macular disease, but I just wanted to briefly cover it for the biological context of my project. So age-related macular degeneration is designated as one of the major blinding diseases in the world, and its late wet form is characterized mainly by the presence of choroidal neovascularization. And now this is when blood vessels sprout from pre-existing vessels that would normally provide regular blood supply to the eye, but they break through into the retinal layers where they shouldn't be. And these newly formed vessels are poorly formed and have a tendency to leak or hemorrhage. And it is this leakage of blood or fluid that can lead to vision loss. So in these figures on the slide, we have a cross-section of the layers of the retina. And this pink rectangle here represents the choroid, and this forms the normal vasculature to the retina. But during neovascularization, blood vessels break through the Bruch membrane, which is this very thin pink line shown here. So I've got this arrow here. And the Bruch membrane is a barrier between the neurosensory retina and the blood vessels. And the neurosensory retina refers to the portion of the retina that is normally avascular, so shouldn't have any blood vessels, and the portion that responds to light stimuli. Then on the right, we have some real photographs of a healthy eye here, and then an eye with wet AMD. And the images show the tissue of the eye and its blood vessels. And these dark purplish spots show areas of hemorrhage in the eye with wet AMD. As I'm sure a lot of you are aware, current treatments target this unregulated growth of blood vessels using anti-VEGF therapies. VEGF is a large protein heavily involved in the body's processes of making new blood vessels. And the anti-VEGF therapies currently available are also very large molecules due to their need to bind to VEGF. Because of this, they must be formulated and given to the patient via injection, which obviously can be quite uncomfortable, painful, and as well as requiring hospital visits. Anti-VEGF drugs have also been associated with resistance issues as well as unwanted side effects. Hence, there is a gap in the market in terms of drugs for wet AMD, especially perhaps something that's a bit of a smaller molecule drug that maybe could be administered differently, such as maybe in an eye drop formulation, and maybe one that binds to, say, a different important protein in the process of new blood vessel formation. So this leads me on to finding this new drug. Historically, many plant species have been found to contain medical compounds. So a couple of examples you may have heard of are the anti-cancer drug Taxol. This is a very complex compound derived from the bark of the Pacific yew tree. Another example is morphine, derived from the opium poppy. So this project started years ago because researchers took an interest in plant species belonging to the Asparaginaceae family, also known as the asparagus plant family, as they have been known to have been long used by traditional healers in Eastern and Southern Africa. So these plants are made into decoctions and infusions and are known to be used holistically to treat arthritis, inflammation, diabetes, bacterial infections, and also to aid in wound healing. So on the slide, we have an image from the World Health Organization showing an African market where dried plant species are sold for uses in traditional medicine. And on the right, we have two plants belonging to the Asparaginaceae plant family that we're going to discuss next. So why do these plants have a healing effect? Well, Homoisoflavonoids are a class of biologically active compound made by these plants. And they were first discovered after extraction and isolation from a plant called Eucomis bicolor in 1967. So this image on the left shows the Eucomis bicolor plant sprouting its green leaves, and it is well known as the pineapple lily plant. So pure compounds like homoisoflavonoids are able to be isolated from plant material, first through extraction. This usually involves cutting up material or turning it into like a mulch, for example, the dried bulbs or leaves of the plants. And then this broken down material is then left to mix with a chemical solvent, and this dissolves the compounds in the plant, and it sort of leaches them out of the plant. Obviously, this produces quite a complex mixture of plant metabolites, but this mixture is then separated over many, many steps in the chemistry lab to eventually produce a pure compound. So this chemical structure here on the screen shows eucomin. This is the first documented homoisoflavonoid from Eucomis bicolor. I understand some people might not be some people might not be familiar with chemical structures like this, but just basically each corner or point here represents a carbon atom. So we have basically a 6-membered ring of carbon atoms here, and then we've got the O's representing oxygen and the H's representing hydrogen. Not all homoisoflavonoids look like eucomin. There are many different types, but the ones we're going to talk about today all feature this carbon skeleton as shown by the colors on the screen. So we have these 2 rings here, the blue and green, with the oxygens, and they're always linked by this one little carbon here, and then to another ring, and we call these rings A, B, and C. Since the discovery of eucomin, homoisoflavonoid compounds have been shown to exhibit a broad range of biological activity, activities such as anti-cancer, anti-inflammatory, antifungal, as well as anti-angiogenic properties. If you're unfamiliar with the term angiogenesis, this means the sprouting of new blood vessels. So anti-angiogenic means that these compounds have been shown to be able to reduce the sprouting of new blood vessels, which could obviously be ideal for treatment of wet AMD in the correct circumstances. So how do we actually go about testing these compounds in real life to see if they could potentially be developed into drugs? Well, this is done in a biology lab where they culture live human cells. So on the left of the screen here, there's a picture of me at our collaborators' lab, and this was at the Indiana School of Medicine. As you can see, I'm in my lab coat, and I'm performing what is called a cell-based proliferation assay. Proliferation refers to when cells reproduce and increase in number through growth and division. So obviously, if we're having new blood vessels form in the macula, this is what will be happening to those cells that line the blood vessels during neovascularization. Obviously, they'll have to be growing to produce that new vasculature. So the first type of cell we test against are called retinal microvascular endothelial cells. Long-winded, but this just means the cells that line your retinal blood vessels. So how do we perform these assays? I'm sure this might not be familiar to most, but on the right-hand side of the screen, there's sort of a see-through plastic plate with lots of little cylindrical little cups on it. These little cups are called wells, and in each well, we plant some live human retinal endothelial cells. Then we add our test compounds, so our homoisoflavonoids that we've isolated, and these are added to these wells at different concentrations. And then after a period of time, we use a color indicator that tells us if the cells have multiplied, proliferated, or if the compound has prevented the cells from multiplying. As you can see, this pinky purpley colors on the screen, this would be the type of thing that we would be looking at. And essentially, the indicator goes from blue, which equals no cells have multiplied, to red, which means lots of cell multiplication has happened. So these purpley colors and lighter purple are like a middle ground. And so what we're looking for is compounds that stop these cells from multiplying, because they'd be interesting to us, because this means they may be able to slow down the growth of new blood vessels involved in wet AMD. So to put this into context with our previous research from the group, here's another homoisoflavonoid on the right-hand side of the screen. As you can see by the colors, we've still got that scaffold, that classic structure that I discussed earlier, but there's a couple of different chemical groups. This homoisoflavonoid was extracted from Lettoboria ovatifolia, minced that, also known as the flat-leafed African hyacinth, shown as that purple flowering plant on the left-hand side of the screen there. And in previous research, this compound was isolated, identified, and shown to inhibit the growth of human retinal microvascular endothelial cells at very low concentrations. So where does my project and my work actually come in? Well, as you can imagine, there are many factors for a successful drug or drug-like molecule that need to be considered before they can be taken into animal or human models. So you want to look at a range of different compounds with a lot of different groups, many as we possibly can. Obviously, first and foremost, what we're looking for is efficacy of the compound. But then there's something called selectivity that needs to be considered. This means we're asking, are the compounds only having an effect on the cell or tissue type that we need them to, or are they having an effect in tissues all over the body? Because this could obviously cause side effects in places where we don't want the drug to be acting. There are many other factors, including solubility in blood, if it was taken orally, how it would be expected to cross barriers within the body, how it would be formulated, and the list goes on and on. But naturally derived homoisoflavonoids are obviously limited to what a plant can make. And when we isolate them, we don't actually know what we're going to get out. So this means that there are only a handful of chemical groups that we find attached to the homoisoflavonoid skeleton. Not to mention that they may be present in very low concentrations in the plant, meaning they can be quite difficult to isolate from the plant material. The project I was involved in, funded by the Macular Society, was to actually synthesize a range of different homoisoflavonoids with different chemical groups on the skeleton, such as well-known moieties used in other drug-like compounds and other non-natural groups. So on the chemical structure shown on screen, the colored dots represent the positions where I'd put different chemical groups and design different molecules whilst maintaining that homoisoflavonoid basic structure that is shown in black. So in terms of the synthesis, we basically aim to make compounds from readily available starting materials that you basically get from like a catalog, and you want to do this in as many steps as possible. But when you're making new compounds, and these are compounds that haven't been made and published before in the scientific literature, it's sort of like following someone else's recipe, but you've got different ingredients. So as you can imagine, sometimes things don't go as you expect them to. So the beginning part of my project, a lot of it was exploring different ways to make these compounds from scratch. But at the end of the project, we have ended up with 48 synthetic homoisoflavonoid compounds, most of which are new to the scientific literature. 26 of these compounds were shown to be active against the proliferation for the growth of human retinal endothelial cells. And when we say active, we mean effective against the growth of the cells at levels below our baseline concentration. And our baseline concentration is 100 micromolar. Don't expect anyone to understand what that means, but to put it into context, if we had a pint of water, to get that concentration of salt in our water, we would only need to add about 3 milligrams of salt, which is obviously quite low concentration. So we have this baseline as when we're looking for druggable compounds, we want the pharmaceutical effective dose to be as low as possible, as obviously we don't want to be overloading our bodies with mass concentrations of drugs. And this also rules out the least active compounds at the same time. So an example of one of the 48 synthetic compounds made is shown on the right-hand side of the screen. We can see that this, the 3 hexagonal ring structure is still intact, but attached at this position here, highlighted in blue, we have what's called an amino acid residue. If you're unfamiliar with what an amino acid is, essentially, they are chemical building blocks that make up proteins in our body. So this little schematic here just describes that. So each amino acid is a little circle, and then a protein is when we've got all of these joined together in a chain. So we don't find these types of groups on naturally occurring homoisoflavonoids, but they are often added to drug-like molecules to increase bioavailability. So this means the ability for a drug to be absorbed and used in the body, to decrease toxicity, and they may even be more active than their non-amino acid counterparts because obviously the protein's made up of amino acids. So if it's got an amino acid on it, the compound might bind better to proteins of interest within the cell. So this is the type of graph that we would use to depict the effect of the compound. The effect of the compound is having on the growth and replication of the retinal cells. So we have two axes. The vertical or y-axis here refers to the percent the cells were able to proliferate or grow compared to a control. So for example, a control would be a test that had no compound in and the cells were just allowed to proliferate as they would without any inhibition. And then on the horizontal or x-axis, we have a concentration of our compound. It just looks a bit funny because it's in a logarithmic scale. And we have a sigmoidal shaped curve. And as you can see, as the concentration increases from left to right of our compound, the percentage proliferation decreases. So it says GI50 2.5 micromolar here. What does that mean? The GI50 value of any drug-like compound that we're looking at basically means the concentration of that compound required to decrease the level of the cell proliferation by 50%. So how much compound is required to reduce the cells replicating by 50%. Obviously, micromolar I won't be familiar with, but put it back in our into our salt analogy, we would need to add less than 2 grains of salt to a pint of water to get this concentration of salt in the water. So obviously this compound is effective at reducing the rate of retinal endothelial cell proliferation at quite a low dose. So what's next? Well, of course, these results are promising, but there's much more work to be done. More hurdles to get over before we can say if these compounds would be effective as drugs for wet AMD. Firstly, we have to test to see, does this compound stop any other cell type from proliferating, or is it selective to just the cells involved in the disease state? The more selective, the better to reduce unwanted side effects, but there can often be a balance with this. So the synthetic compound just discussed was found to be inactive against the proliferation of a retinal pigment epithelial cell line. This is very important as these cells in the these are cells in the retina that help you to actually see. So we don't want to be stopping them from functioning. And obviously they're very close in space to the cells in the disease state they're in your eye. So it's very important these compounds do not affect these cells whatsoever. This compound was also shown to be inactive against an ocular melanoma cell line, so cancer cell line, which may seem strange, but testing against cancer cell lines helps us to ascertain whether a compound is actually just toxic rather than affecting a process within the cells, as toxic compounds will sort of just destroy most cell lines. So it's good that it was showing this selectivity. There are also more tests that can happen outside of living animal that are currently being done at our collaborators' lab with the compound just discussed. And these are called migration and tubule formation tests. So during the process of new blood vessel formation, or angiogenesis, the cells that make the new vasculature have to proliferate from existing vasculature, so grow and replicate, but they also have to move into the space where the new vessels will form. So when the new blood vessels break through the Bruch membrane in wet AMD, there was obviously no blood vessels there before. So the cells need to move and migrate. To make new vessels, they also need to join together to form like tube-like structures so the blood can flow through. So that make up the vasculature. So we're looking for a compound that stops both of these things from happening to the blood vessels in the retina to prevent wet AMD. So basically we can test for the compound's effect on the cell's ability to migrate and to form these types of tubes. So on the slide here, we have these two microscope images, and this shows a migration test assay. The little white dots are actually single cells, and as you can see on this left-hand image, there's a black rectangular section where no cells reside, where they have been sort of scratched away. And this is how we test the migration of the cells into an empty space. Test compounds are added to the cells at different concentrations, and then we look through the microscope again at certain time frames to assess how well the cells have migrated back into this empty space. And as we see here on the right, there's a lot more white dots where this empty space was before. So this is an example of cells migrating back in. The tube formation is done in a similar way. This is where cells begin sort of on their own on the plate, and it's assessed how well they join up together over a period of time. So here on the slide, we have microscope images of tubes being formed. I'm sorry if they're a bit hard to see. It's often hard to focus the microscope correctly, and they're always in this like gray color. But basically, the first picture just shows the cells as sort of little dots, and then they progressively join up together to sort of a more vascular, like web-like shape. So basically, if our test compound comes back and shows to inhibit the migration and tubal formation of retinal microvascular endothelial cells, it may be more likely to be effective inside of a living creature. And the first thing that would be done is usually mouse studies. So at the end of this project, what have we achieved? Well, we've got an alternative way of synthesizing these compounds from scratch, not from the plants. We have tested 40 plus novel compounds, compounds never been published before, for activity against human retinal endothelial cells. This massively helps us to further assess the relationship between the chemical structures of these compounds with the effects we observe them to have in the cells. We have incorporated non-naturally derived chemical groups and identified 26 active compounds. And the most active compounds are currently being tested for migration and tubal formation effects. So what's next? We haven't published this work yet, as I said, but that should be happening in the next few months. Another supervisor of mine at the University of Surrey, Dr. Welligan, has obtained a grant to synthesize more derivatives. And I know Professor Mulholland is very keen on the research continuing. So of course, there is still much work to be done, but I hope you find it exciting that there is research constantly going on to try and find more solutions to treating macular disease. And hopefully we can uncover a new biological target to reduce this blood vessel formation, eventually causes vision loss. To finish up, I just wanted to say a massive thank you to the Macular Society for funding the project. Their generosity, alongside the support from numerous other grants, such as the Royal Society of Chemistry and Santander, has allowed us to take part and present this research at numerous natural products conferences abroad. So here on the left is me last year in Krakow in Poland, presenting at the International Congress of Natural Products Research. I presented the year before in Dublin to an audience at a conference with the Society for Medicinal Plant and Natural Products Research, which is also known as the GA. And then here on the right, I'm presenting a poster this time at a GA conference in 2022. It's been fantastic to be able to get the word out about this research, as well as bring awareness about macular disease to other younger scientists who may have never heard of it or been affected by it. And as I mentioned earlier, I was lucky enough to learn the cell-based science with our collaborators on a placement, which was an amazing experience and learning curve. So I just wanted to say thank you again and hope you enjoyed the talk.
Thank you so much, Elisha. And obviously that funding from the Macular Society was only possible because of the donations that we received. And I'm sure many of the people on this call will have been people who've donated or raised money for the Macular Society. So thank you everyone who contributes to our work and our research programme and allows us to fund fantastic work like Elisha's done. Really, really interesting to hear that. I can see some questions are coming in already, but please do use the chat functionality if there's any questions that you'd like to ask Elisha. One of the first questions was answered in the chat by Dulcie, but I might just build on it a bit. That someone had asked whether there's studies on humans yet, and I understand the answer is not yet.
No.
And I guess the natural next question is how far away do we think we might be from being able to try some of these studies on humans? And welcome again, Dulcie, who'll be joining us for the Q&A too.
I would just like to add to the eye drop part and getting onto humans. My another super of mine, Sian Shrikov, was telling me about her research that she'd done on bull's eyes corneas, where they were testing the solubility of the compounds and if they would actually dissolve through, and they got some quite promising results with that.
Yeah, do we have any sense how far away, you know, if all goes well, how far away this might be from being tested in humans?
Well, I would say in the next week, we're going to be evaluating our most active compounds, but while we write up our final report. And then it's mouse model studies of the best occupying. And then after that, funding permitting to go on to human studies.
So some way off yet.
Unfortunately.
Yeah, but you know, this is how work progresses, isn't it? It starts early and it builds in stages. But it's great to see a new area being explored. Really fantastic. So I'm just going some of the other questions. What's been the most exciting or surprising part of your research journey so far?
A hard one. I think how much my skill and interest has grown over my research journey. I think that's I don't know if it's surprising, but I didn't expect it to look back on my first year and be like, wow, like I wish I knew what I knew then.
Yeah. No, interesting.
There's a question about how the compounds were sourced. Well, originally we were getting them from plant material in Southern Africa. Then it became difficult to get plant material from there with new legislation. So we were buying bulbs in bulk from bulb suppliers in the Netherlands. So a lot of our compounds came from there. And then we realized that if we were really going to be serious about this project, we need a better way of doing it. If you get them from the bulbs, they come in really complex mixtures and take a really long time to separate and purify. But by synthesizing them, we could get around those problems and get a purer compound much quicker and far more cheaply.
So the compounds are inspired by plants, but not actually from.
Correct. Right? Although within this project, we've also, I think, put 15 more plant-derived compounds into the screening, and we've got some nice results there as well, which then leads Elisha to know which direction she should go in the synthesis.
Yeah. The discussion so far has been on wet AMD, and the question is whether this might have potential for dry AMD or indeed other forms of macular disease.
So because the main activity that we see is the anti-angiogenic activity, which is the reduction of the blood vessel formation, that would be mainly for dry AMD, because as we know for wet AMD, we know for dry AMD, it's mainly the start of it's that drusen formation, and we don't know what the effect of these compounds is on drusen formation because we haven't seen them in an eye yet.
Yeah, thank you. And there was a question which I think you'd answered in the chat, Dulcie, but how would you see this plant therapy being administered potentially? Just for people who haven't seen the chat, if you could.
After talking to our colleagues in Kingston, they're very keen to go the eye drop route, which would be the ideal route, make life so much easier. I mean, this treatment would be so cheap compared to the ones currently on the market, because once you've got your synthesis going, it's very cheap to make.
Yeah, fantastic. It's an interesting question. Thank you for the brilliant work. In the meantime, should we eat salads of the various plants?
I'm not sure about that one, but a lot of factors influencing AMD are based on oxidative stress. So anything antioxidant, I would recommend taking antioxidants, have a little research about what antioxidants might be good. I'm just doing that. Maybe not the African lily for dinner.
In general, eating salads is going to be a good idea, isn't it? And we
I
certainly
mean, eating salad is always a good
recommend
idea.
people to eat, plenty of variety of fruit and vegetables and eat the rainbow. So, yeah, Dulcie, did you want to come in on that one?
Well, flavonoids are well known as good antioxidants, and they only differ from our homoisoflavonoids by 1 carbon. Our homoisoflavonoids have got 1 extra carbon to flavonoids, and flavonoids, of course, are in green tea, they're in red wine, lots of fruits. So yes, not a crazy idea to keep eating the fruit.
The scientific endorsement for red wine was that.
Absolutely.
But yeah, salad's certainly not going to do anyone any harm. And a question about sort of moving away from nutrition on AREDS2 supplements and whether you could see this having an impact on that in any way if the combination of AREDS2 supplements and these homoisoflavonoids, could they interact in any way, or is it too early to say that?
I think it's too early. It's not something we've really investigated.
Yeah.
Okay. And sadly, when we first, this research was, when we first founded, we patented with the University of Indiana and Kingston University, just to protect it. And what was I going on to say there? It appeared in the newspaper, and we had people from around the world volunteering themselves for research. And it was very sad to tell them that it's not at that stage yet.
Yeah. But for people who haven't seen, there was a link posted by my colleague earlier for, what we're talking about today is too early for human trials, but we do have a research participant database you can find on our website. So that's somewhere you can register to get involved in all sorts of research. So if you're interested in participating, please do join that. And that's not just clinical trials, that's also social research and all sorts of different things. Going back to the eye drops kind of mode of administration, someone's asked how would the drug get to the retina, and why can't the current anti-VEGF drugs be administered by eye drops?
So the current anti-VEGF drugs are thousands and thousands of times the size of the molecules that I was showing today. So they're basically large, proteins, so that can't dissolve through layers like your cornea. It can't dissolve through places like that. It has to be very specific to actually be able to dissolve through. But as I did mention, there was some promising results where it looked like these type of compounds would dissolve through the cornea. But obviously they would then be in that humorous part of the eye, but they'd have to get to the back of the retina.
Yeah. Okay. Interesting. So the size of the molecule being crucial. I'm just looking. Lots of people want to get involved in the research. I think we've covered that. We've had a question whether you have a link to the research paper, and I think you said it's not published yet.
Not yet.
We've also had questions here about whether we've got a link to a paper on eye drops. That was just in consultation with our colleagues at Kingston. I don't think they've published anything on that.
Yeah, but I'm sure once it is published, I'm sure we'll share news on that in our kind of normal communications channels. So keep an eye out, and things like SciView and our e-newsletter will let people know. There's a question you haven't talked about dry AMD. So we did cover that earlier, but this is specifically for wet AMD because it's to do with growth of the blood vessels. So that's why you've been focusing on this. I'm just scanning through the questions. Lots of people wanting to take part, which is always understandable. Are there any different questions? I mean, just to go back to real sort of basics, why these compounds? What led you to go down this?
I've just been reviewing a big grant proposal for some Saudi university, and they were going to look at some plants, and they were claiming they were going to do all sorts of things, which I said, well, I don't think that's quite correct, because natural products is regarded as like a fishing expedition. You take a plant and you never know what you're going to get out. You never know if compounds are going to be active, toxic, or whatever. And this happened totally by chance. We had looked at a Southern African Scilla species, got a compound out, and Tim Corson, who was in Indianapolis at the time, now moved head of pharmacy at University of Toronto, he emailed me, and I didn't know him, saying he saw we'd published this compound. He'd heard there'd been a Chinese paper saying that these types of compounds had anti-angiogenic activity, and could we possibly send him some of our compounds to put through his screen, which we did, and we found that one that Elisha put up to be really quite active. So it was total chance that we worked. We weren't looking for the compounds to treat this condition. Tim just contacted us. I believe Tim did a webinar in 2022. So some of you might remember him. So it's luck, finding new drugs very often.
It's not the most reassuring answer, but it's obviously the truthful answer. I guess it highlights that importance of collaboration, and so much of science is about those collaborations, isn't it, internationally and across universities.
Absolutely.
And across disciplines as well, I guess.
Yeah.
Absolutely. As a chemist, we don't have screens and things in our laboratories. We can synthesize them or tell you what the structure is, but we need the biologists to work with us to do the cell-based studies and the mouse model studies, etc., and carry on with it. So all collaboration.
Yeah. Right, just having a look. Dulcie mentioned, or we're going a bit off topic, but let's go for it. Dulcie mentioned flavonoids in red wine being a good thing. Are there any risks with alcohol causing inflammation or rupturing of the blood vessels?
Oh, well, I'm not a pharmacologist, I'm afraid. I'm a chemist. I wouldn't know. Everything in moderation.
Everything.
Yeah, I think too much alcohol would definitely cause oxidative stress, which would contribute to bad symptoms rather than good.
So everything in moderation. You've heard it here first. Good, solid advice. Would cataract surgery? Oh, sorry. As people ask questions, it scrolls down. Would cataract surgery affect the drops going through the layers of the eye to the retina? So,
that sort of study would be way down the line, I think.
Yeah.
Yeah. Here's an interesting question. Do we know what type of characteristics these compounds impart to the plants?
Now that's a very interesting one. When somebody asked earlier whether we should be eating these bulbs, my answer would be absolutely no, because some of them coexist with cardiac poisons, cardenolides, etc. So eating them is absolutely no. So what do? We know that perhaps those cardenolides protect the plant against sheep and cattle, etc., eating them in the wild. And in fact, that's how these compounds were originally, why these plants were originally looked at way back, because sheep were being killed by eating some of these plants. But the homoisoflavonoids themselves are generally seem to be quite non-toxic. And they seem to be, well, we know from a separate project, which has been shelved because this has been more important, they've got very good anti-inflammatory effects. So why a plant would produce compounds with anti-inflammatory effects, we don't quite know. So the reason why homoisoflavonoids occur in plants is still a mystery.
Fascinating. Yeah, really interesting.
They are colored yellow, but in a bulb it doesn't really matter what the color is because you don't see it underground.
Yeah, and the person responds, if we did, then maybe we could reverse engineer plants with specific characteristics to identify the unique compounds.
But as you say, we found the synthesis to be cheap and relatively straightforward. So we could, based on Elisha's work, synthesize pretty much any active one that we could find in big amounts very cheaply.
There's a comment on the sort of method of delivery. While smaller molecules diffuse more readily, they are not directed by diffusion, whereas injection makes sure it's delivered inside the eye, and therefore a higher access to the retina is obtained.
Sure.
Avoiding injection is desirable, but would injection be considered for such drugs?
Oh, absolutely. I think further down the line, all methods of delivery would be investigated and compared.
Yeah. I'm just going through. I think we've answered most of the questions.
Perhaps your audience would like to hear about a very unrelated other patent.
If you think it could be interesting, please share. Yeah.
Well, see, we've been talking about wine, and this is again another one just by chance. We're doing field trials at a vineyard near Guildford. And this was another just chance discovery where we were part of a massive EU project looking at waste across Europe of bark waste, because the people needed the wood, but they just burned the bark or discarded. And so we were working with an organic farming institute in Switzerland who had a whole lot of plant pathogens. And we isolated about 150 compounds, and they tested these against these plant pathogens. And they came up with two of our compounds were really good against a disease called downy mildew. And downy mildew can really wipe out a vineyard in a wet year. And at the moment, copper products are the only things that you can use to prevent this disease. And so even in an organic vineyard, you think you're buying organic wine. It's not really organic because they have to use copper to get a crop. And the regulations allow for a certain amount of copper, even in organic wine. And so we've had very successful field trials at this vineyard and in Southern Europe as well. And again, it was just a chance discovery from looking at a plant. So you never know what you're going to get out of a plant.
I love the fact that your research encompasses macular disease and vineyards. It's quite far-reaching. This is an interesting question. Could AI help in designing molecules that target receptors on the newly formed vessels in AMD?
Sorry, which is that one?
Could AI help in designing molecules that target receptors on the newly formed vessels in AMD?
Absolutely. Now, Elisha made this big library of compounds, and if you put you got your structures, and then you've got your results of your screening, you put them all into AI, and AI will pick up which positions are important for modification, etc., to make the most effective drugs.
Is this something that's being done?
Well, Elisha's done it using her own brain at the moment, but feeding it into such a system would certainly speed up the process.
Yeah, there is quite complicated systems. I mean, it's called QSAR, isn't it? It involves calculations and things, because obviously the structures I showed you, they're not actually stationary in real life. They move when they're within a biological setting. So it's a bit more complex, especially if you know what the target is, because then you know what kind of chemical groups are inside the center of that target, and then you know how a certain molecule can move and how energetically that would be the most stable and things like that. So it's a bit more than just saying, oh well, that's good, that's, you know, like it needs like calculations involved in quantum chemistry and things like that.
Yeah. Fascinating. Just scanning through questions if there are any we've missed. I can see a few people sort of ask quite personal questions about their own treatment. And what we'd advise is that if you get in touch with our helpline, I'll just read out the number for people who can't see the chat. So it's 0300 30 30 111. So 0300 30 30 111. So if you give them a ring tomorrow, they can talk you through, information about your own condition and your own treatments, because obviously we're not in a position to do that tonight. Or you can also email info@maculasociety.org. So that's info at macularsociety.org, and we can help out with any individual questions people have. So yeah, I think I'll just say thank you again so much to both of you. It's been fascinating hearing about your research, Elisha. Really, really interesting, fascinating idea that we're exploring. And thank you for everyone for joining us and for all of your questions. Really good to kind of be with you all here on Tuesday evening. And just a reminder, our next webinar will be on Tuesday the 20th of May, again at 7 p.m. We haven't announced the speaker yet, but I'm sure we'll have a fantastic speaker, so watch this space and we'll let you know who's speaking there. And yeah, thank you again to our special guests, Elisha and Dulcie. And all the best, everyone. Have a lovely evening.
Thank you.
Thank you.
Bye.